Hi and welcome to my channel. Considering a request from some friends, I'm planning to do a series of videos on the subject of pathology. Pathology as such is a very vast subject, also a very key subject in the medical curriculum. It is the first subject which exposes the medical student to what is abnormal or to the study of abnormalities or to the study of diseases, the treating of which makes their career. In that sense, it's a very key subject and also a very vast subject. The scope and depth of my videos here is not about covering all the finer details of this very vast subject. The scope of my videos are only discussing the basic outline or the basic concepts of the important topics from an exam point of view. I will be discussing only the basic outline and concepts. In order to understand the finer details and nitty-gritties, the student will have to do further reading. The aim of my videos here is to make that further reading easier by explaining the basic outline and concepts of important topics. Okay, come on. Together, let's start exploring the different dimensions of pathology. Let's start with what is pathology. The word pathology is derived from two words, pathos, logos. Pathos means suffering, logos means study. Pathology is the study of suffering or the study of diseases. It is concerned with the study of changes in structure and function of body in disease. Anatomy is concerned with the study of the normal structure, and physiology the study of normal function. Here, pathology is the study of changes in structure and function of the body in disease. Okay, let's move into the subject proper. The first topic we will be discussing is cell injury. As you know, the basic unit of human body is cell. So study of any disease or any abnormality or any change in structure and function in a disease should also start from the basic unit, that is the cell. How a particular condition brings about changes in structure and function of a cell, and what are those changes in structure and function at the cellular level. I have divided the discussion on cell injury into two parts, and this is part 1. Okay, let's start, and let's start with the basic question: what is cell injury? Cell injury is the effect or response to a variety of stresses a cell encounters, which results in changes in its internal and external environment. Cell injury is the effect or response of the cell to a variety of stresses that particular cell encounters, which results in changes in its internal and external environment. And this response can take two forms. Either the cell may adapt to that particular stress or it may die. Just like as humans, when we face an adverse situation, either we adapt to that particular situation or we may succumb. The same way, the cell, when it encounters a stress, it may adapt to that particular stress by undergoing certain modifications which help it to adapt to that stress, or it may result in a permanent injury resulting in the cell death. The first one, that is, the adaptation is mostly reversible, but cell injury is permanent loss of cell. First, let's discuss the cellular adaptations. The adaptations can take one of these forms: atrophy, hypertrophy, hyperplasia, metaplasia, and dysplasia. First, let's see what is atrophy. Atrophy refers to a reduction in size of the cell. The overall size of the cell reduces. It may be physiologic or pathologic. Example of physiologic atrophy is the atrophy of thyroglossal duct, which happens during the embryonic period, and example of pathologic atrophy is disuse atrophy. That is, when an organ is kept unused, it undergoes atrophy. Yes, you are right. So keeping your brain unused for a long time is not advisable. And another example is denervation atrophy, when there is interruption in nerve supply to a particular limb. For example, a paralyzed limb, it can undergo atrophy. You must have seen it, a paralyzed limb which is also atrophy. Coming to hypertrophy, hypertrophy is the increase in size of cell. This again can be physiologic or pathologic. Physiologic, the example is, of course, the hypertrophy of muscles in exercise. Pathologic example is the hypertrophy of cardiac chambers in some hemodynamic overload like blood pressure. Coming to hyperplasia, hyperplasia refers to increase in number of cells. Remember, hypertrophy is increase in size of cells and hyperplasia increase in number of cells. In hypertrophy, the size of the organ increases, but that is due to increase in the size of the cell. The number doesn't change. If there are 4 cells, the cells remain 4, but the size of each cell increases. On the other hand, in hyperplasia, the increase in size of the organ is due to increase in number of cells. If there are 4 cells, they multiply to 8. This number of the cells is increased. This again can be physiologic or pathologic. The example of physiologic is the hyperplasia induced by hormones. Example, female breast during puberty or pregnancy. Pathologic hyperplasia can also be hormonal. Example, endometrial hyperplasia following estrogen excess. But the pathologic hyperplasia is more serious because it can lead to cancer. Metaplasia refers to change of one type of cell to another. For example, a columnar cell transforming into a squamous cell. There are basically 2 types of metaplasia: epithelial metaplasia and mesenchymal metaplasia. The name is self-explanatory. Epithelial metaplasia is one type of epithelium transforming into another under stress. Here, mostly the change is from a more specialized epithelium to a stronger but less specialized epithelium like squamous in order to ward off that particular injurious stimuli. Classic example is the epithelium of respiratory tract in smokers. In smokers, the normal pseudostratified columnar ciliated epithelium of the respiratory tract transforms into a more strong squamous epithelium. Another example where squamous epithelium transforms to columnar is at the lower end of esophagus in the condition of gastroesophageal reflux in order to withstand the attack of the acid. Mesenchymal metaplasia is one adult mesenchymal tissue transforming into another type of adult mesenchymal tissue. Example is myositis ossificans, where there is formation of bone within muscle, and this happens due to trauma. Coming to the last one, there is the dysplasia. Dysplasia refers to disordered cellular development. That is, the cell growth or the cell multiplication is disordered. This mostly is preceded by hyperplasia. In hyperplasia, the cell number is increased. Here, in addition to increase, there is also disorderly growth. So it is called atypical hyperplasia. Dysplasia mostly occurs in epithelial cells. And what are those disorders, or what are the features of this disorderly growth? That we will see now. The features include increase in number of layers. In multilayered epithelium, there is increase in number of layers than what is normal. Number 2, there can be disorderly arrangement of cells within these layers. The cells are not properly arranged. Number 3, there can be loss of basal polarity. Usually, the nucleus lies near the basement membrane, but here in dysplasia, the nucleus is on the opposite side, that is away from the basement membrane. This is called loss of basal polarity. There can be cellular and nuclear pleomorphism, that is variation in shape. There can be increased nucleocytoplasmic ratio. The usual nucleocytoplasmic ratio, that is the ratio of nucleus to the cytoplasm, is usually 1:5, and here it can be increased nucleocytoplasmic ratio. There can be nuclear hyperchromatism, and there can be increased mitotic activity. These are the disorders seen in the disorderly growth called dysplasia. And this dysplasia may progress into carcinoma in situ or invasive cancer. You can see this as a linear process: hyperplasia followed by dysplasia, dysplasia leading to carcinoma in situ or even invasive cancer. We will discuss about carcinoma in situ and invasive cancer in some other video while we discuss neoplasia. For the time being, remember what is dysplasia. It is disorderly growth. Okay, now coming to cell injury, which may lead to cell death. The cell injury can be reversible and irreversible. When the stress or stimuli encountered by the cell is something which the cell can adapt to, it undergoes the various adaptations that we discussed earlier. When the stimuli is more severe or more persistent, the cell gets injured. And this injury can be reversible or irreversible depending upon the persistence and severity of the stimuli encountered by the cell. Before discussing about the reversible and irreversible cell injuries, let's see what are the stimuli which can cause these injuries. The cell injury can be caused by hypoxia or ischemia. Even though both seem similar, there is a fundamental difference between hypoxia and ischemia. Ischemia refers to the interruption in or the total cutoff of blood supply to the cell. So the cell is derived of both nutrition and oxygen. Hypoxia refers to certain conditions where only the oxygen supply is disrupted. Example, anemia, where the oxygen carrying capacity of the blood is reduced. So in hypoxia, there is deficiency in supply of oxygen, and in ischemia, there is deficiency of both oxygen and nutrition due to total cutoff of blood supply, and both these cause cell injury. We will discuss this a little more in a short while. Before that, coming back to other agents which can cause cell injury, these include physical agents like mechanical trauma, thermal trauma, electricity, radiation, etc. Chemical agents like poisons, acids, alkalis, pollutants. Biologic agents, of course, the most heard term these days, novel coronavirus, virus. It is also a biological agent which can cause cell injury. Apart from virus, there are other biological agents like bacteria, fungi, parasites, etc. Cell injury can also be immunologic. Cell injury can be caused by body's own immune system. Immune system is our defense against external agents, but this immune system is a double-edged sword. So sometimes in the process of attacking foreign agents, it can also damage our own cell, causing cell injury. The other causes of cell injury include genetic derangements and nutritional imbalances also. Now that we have seen the causes, let's move on to the mechanism of cell injury. Before going into the mechanism, let's also see the factors which affect this process. The factors which affect this process can be grouped into two: the factors pertaining to the stimuli in question and the factors pertaining to the host, that is the cell in question. The factors pertaining to the stimuli include the type of stimuli, whether it is hypoxia, ischemia, or biologic agent, or physical agent, or chemical agent, or whatever, and the duration of the stimuli and the severity of that stimuli. The factors pertaining to the host includes the type of cell, what type of cell it is. For example, A skeletal muscle cell can withstand hypoxic injury for a long time than a cardiac muscle cell. Okay. So what type of cell it is, and also the adaptability of the cell. Some cells are more adaptable to certain injurious stimuli than others. So the host factors include the type of cell and the adaptability of the cell. Now coming to the mechanism of cell injury. The first thing which happens is the depletion of ATP or the decreased generation of ATP. This occurs due to deficient supply of oxygen or glucose or both. Here there is a difference which we discussed earlier while discussing hypoxia and ischemia. In hypoxia, only the oxygen carrying capacity is reduced, and so the oxygen is deficient. So the cells can switch to anaerobic glycolysis. You must have learned about aerobic pathway, anaerobic pathway, and all those in biochemistry. Let me not go into it. When only oxygen is deficient and glucose is available, the cell can switch to anaerobic glycolysis. But as you know already, anaerobic glycolysis is less efficient and only less number of ATP is produced. On the other hand, when both oxygen and glucose is deficient, as in ischemia, where the total blood supply is cut off and there is no oxygen and no nutrition, the anaerobic pathway cannot be activated. Here again, remember another thing, not all cells can shift to anaerobic pathway in the absence of oxygen. For example, the cardiac muscle cells, the neurons of CNS, they are wholly dependent on aerobic respiration. They cannot shift to anaerobic glycolysis. Anyways, whether shifting to anaerobic or not, the point here is the generation of ATP is reduced and there is depletion of ATP. And this depletion of ATP interferes with the generation of phospholipids, which are essential to cell membrane and membrane pumps. So there is defective functioning of cell membrane pumps like sodium potassium pump, calcium pump. Okay. The defective functioning of sodium potassium pump leads to loss of potassium from the cell and influx of sodium into the cell. And you know, there is a saying in India, whoever takes salt should take water. Salt is nothing but sodium chloride. So here also there is influx of sodium into the cell, and this sodium always carries water with it. Wherever there is sodium, there will be influx of water. So this influx of sodium is accompanied by influx of water, which leads to cellular swelling. And now the other pump which can go defective, that is the calcium pump. Okay. The defective functioning of calcium pump leads to influx of calcium. What happens due to influx of calcium? We will come to that in a short while. Now, there is also alteration in the energy mechanism of the cell. We saw that already. When there is deficiency of oxygen, the cell shifts to anaerobic pathway. But the shifting to anaerobic pathway has a side effect. There is accumulation of lactic and pyruvic acid, which are byproducts of this anaerobic pathway. So there is accumulation of lactic and pyruvic acid, which can lead to acidosis. There is also reduced protein synthesis, obviously, and we already saw there is influx of calcium due to defective calcium pump. Okay, this influx of calcium leads to activation of various enzymes like phospholipases. These phospholipases, as the name suggests, degrade the membrane phospholipids. We already saw due to decreased production of ATP, there is decreased replacement of or decreased replacement synthesis of membrane phospholipids. In addition to that, to add insult to the injury, here this activation of phospholipases degrades the membrane phospholipids. The calcium also activates proteases which damage the cytoskeleton. It also activates the endonucleases. The endonucleases, as the name suggests, damage the nucleus. It causes pycnosis. That is condensation and clumping of nucleus, karyohexis, fragmentation of nucleus, and karyolysis, dissolution of nucleus. This influx of calcium also causes mitochondrial damage by collecting in the mitochondria and disabling its function. There is also activation of lysosomal hydrolytic enzymes which are released from the lysosomes because the membrane of the lysosome is damaged, because obviously that membrane is also made up of phospholipids. Yes, these released hydrolytic enzymes are activated by the acidic pH already existing in the cell because of the anaerobic pathway and accumulation of lactic acid, is causing acidosis. These activated enzymes like hydrolase, DNase, RNase, protease bring about enzymatic digestion of the cellular components, completing the cell injury cell death cycle. The dead cell is eventually replaced by masses of phospholipids called myelin figures, which are removed by the scavengers, that is the macrophages. Okay, now that you have seen the mechanism of the cell injury, now let's also see the mediators of cell injury which mediate this process. The mediators of cell injury are calcium and free oxygen radicals. The role of calcium in cell injury we have already discussed in detail. Now we will see about the free oxygen radicals or oxygen free radicals, the discussion about which and their antidotes, the antioxidants, are very much in public discourse these days. Oxygen free radicals are highly reactive oxygen molecules which has an unpaired electron in the outer orbit, which gives it the reactivity. These reactive oxygen molecules are intermediate chemical species formed during the reaction of formation of water from oxygen and hydrogen. The normal reaction of combining of oxygen and hydrogen to form water involves a four-step process with transfer of one electron in each step, and this produces the intermediate species of oxygen free radicals, which include the superoxide, hydrogen peroxide, and hydroxyl radical. And this reaction takes place within the inner mitochondrial membrane. The free radicals cause, number one, lipid peroxidation of cell membranes. As I told you, the free radicals are highly reactive oxygen molecules, and so they oxidate anything that comes across. Okay, they oxidate whatever comes across its way. So they cause oxidation of cell membranes, that is lipid peroxidation of cell membranes. They also oxidate the proteins, the oxidation of proteins, and they also cause DNA damage. These harmful free radicals are balanced by certain substances called antioxidants. The antioxidants can be endogenous or exogenous, and these inactivate the free radicals. Examples of endogenous are enzymes like catalase, superoxide dismutase, and exogenous like vitamins. With this, we will wind up this session. In our next video, we will be discussing the morphology or appearance of different types of cell injury, both reversible and irreversible. So, for more videos, keep my channel subscribed. For further guidance or live interactive classes, do drop a mail to me. Until we meet in another video, thanks for watching.